Welcome to the weekly Neurology Deep Dive.
Thank you for having me.
This program is produced by Amerigavanini. I do want to state clearly up front that this audio is solely for educational purposes. We have absolutely no commercial goals here. This is merely a summary of the literature, and we highly recommend that you, the listener, read the full paper for yourself.
Indeed. Reading the full methodology is always essential for clinical practice.
Exactly. So our mission today is to explore a compelling paper titled Identifying and Predicting Fast versus Slow Parkinson's Disease Motor Progressors Using Clinical and Digital Data. This was authored by Ulberg and colleagues and published in the British medical journal Neurology Open in July 2026.
It is a critical paper for our field.
It really is. And for those of you who just need the practical summary right at the beginning, here it is. Predicting Parkinson's disease progression is notoriously difficult, and standard baseline clinical features are essentially useless for forecasting it. However, by combining a standard clinical assessment with a simple 7-minute smartphone application, neurologists can accurately identify fast-progressing patients from day one.
Which is incredibly important for clinical trials and for routine patient care.
Absolutely. So let us dive into the prediction problem itself. We know that Parkinson's disease progression is highly heterogeneous.
Tremendously so. You can have two patients with identical baseline demographics, same age, same biological sex, same disease duration. One remains relatively stable for years, while the other experiences a rapid decline in their motor function.
And to investigate this unpredictability, the authors analyzed a 96-week digital sub-study. They looked at 98 patients with early to mid-stage Parkinson's disease. These individuals were already participating in the exenatide phase 3 clinical trial across multiple hospitals in the United Kingdom.
That is correct. And in typical trials like that one, the standard method for measuring motor decline is the Movement Disorder Society Unified Parkinson's Disease Rating Scale Part 3.
I mean, that is the gold standard we all use in the clinic.
Exactly. It provides a total score based on specific movements. But the issue is that it only captures a single snapshot in time. A patient visits the clinic every few months, performs the tasks, and gets a score.
Which means we miss everything that happens between those visits.
We miss all of it. A patient might have slept poorly, or perhaps their medication timing was slightly off that morning. Those daily variables introduce significant noise into the clinical score.
And the researchers noted that the overall motor progression rate across this entire group of 98 patients was actually very subtle.
Very subtle. On average, their scores on the standard clinical motor scale worsened by only about 2% per year.
That is a remarkably small signal to detect amid all the day-to-day clinical noise.
It makes running a clinical trial exceedingly difficult. If you are testing an experimental drug, trying to prove that it slows down disease progression, that tiny 2% annual change is almost impossible to isolate using just infrequent clinical snapshots.
So because those traditional clinical snapshots were failing to provide a clear predictive picture, the researchers applied a purely data-driven approach to the longitudinal scores.
They did. They utilized a mathematical technique called dynamic time warping to analyze the full 2 years of clinical data.
Let us unpack dynamic time warping for a moment. How exactly does that function in this specific context?
Well, it essentially compares longitudinal data series by stretching or compressing the time axis. It aligns the underlying trajectories of the disease regardless of the absolute time a patient reached a certain severity.
I see. So it groups patients based on the shape of their clinical decline over time rather than just strictly comparing them at week 12 or week 48.
Exactly. And by using this method, the algorithm identified a completely distinct subgroup hidden within the trial population. It revealed that about 27% of the patients were fast progressors.
About 27%. And their clinical trajectory was significantly worse than the rest of the cohort.
Vastly worse. These fast progressors started with much higher baseline scores on the motor scale. On average, they were about 18 points worse on day 1 compared to the slow progressors.
And their rate of decline over the 96 weeks was also much steeper?
Yes. They worsened by an additional average of about 8 points over the 2 years compared to this slow progressing group.
Now, let me ask about that baseline difference. If these fast progressors were already about 18 points worse on day 1, could a neurologist simply look at their demographic data, perhaps their age or their biological sex, and predict their aggressive trajectory from the start?
You would certainly hope so. The researchers tested that exact hypothesis using conventional clinical categorizations.
And what did the data show?
It showed that conventional clinical methods effectively fail to identify this fast-progressing group.
They fail entirely.
Essentially, yes. When they grouped patients by older age at diagnosis or by male sex, the differences in progression were tiny and not significant.
That is fascinating.
For example, older age only added an average decline of about 2 points over 2 years. Male sex also only added an average decline of about 2 points. Neither finding was statistically significant. The traditional clinical grouping is practically blind to this aggressive subgroup.
Which really highlights the core challenge. We cannot rely on basic demographics to separate fast progressors from slow progressors. If we do, we just end up with a highly mixed trial population.
And a mixed population introduces severe heterogeneity, which can easily ruin a clinical trial.
We will definitely discuss that trial heterogeneity shortly. But first, the researchers needed a way to predict who belonged to this fast-progressing group on day one, since standard clinical features could not do it.
Which brings us to the operational breakthrough of the paper, the smartphone application.
Yes, the digital assessment tool. They use an application that guides the patient through a 7-minute battery of motor tasks. And this uses the standard built-in sensors of a regular smartphone.
Exactly. It utilizes the triaxial accelerometer, the gyroscope, and the microphone. The patient simply completes 7 specific tasks in the clinic.
They record 20 seconds of a sustained phonation into the microphone, then 20 seconds of standing still for balance, then 30 seconds of walking and turning, 40 seconds of alternating finger tapping, a 30-second reaction time test, 90 seconds of resting to measure resting tremor, and finally 90 seconds holding the phone straight out for postural tremor.
7 minutes in total. And the predictive performance difference between this digital approach and the traditional clinical approach is striking.
Let us look at those accuracy numbers. They use the area under the receiver operating curve to measure predictive accuracy, where a score of 0.50 essentially represents a random guess.
Exactly. When the researchers attempted to predict the fast progressors using only the standard baseline clinical scores, the predictive accuracy was about 53%.
So the gold standard clinical assessment was practically guessing at random.
Yes, it was barely better than a coin toss. However, when they trained a machine learning classifier, using the digital features collected from the smartphone during that single day one visit, the accuracy increased massively to about 78%.
An accuracy of about 78% just from a 7-minute digital battery. That is a substantial improvement.
It is a massive leap. The digital biomarkers dramatically outperformed the human clinical assessment.
And looking closely at the data, the models selected specific tasks as the most predictive. Walking, postural tremor, and resting tremor features were the strongest indicators of rapid decline. In fact, the resting tremor features were selected most frequently by the models.
Which is a very interesting clinical finding.
It is, because in routine practice, postural instability and gait difficulties are traditionally viewed as the primary indicators of a severe aggressive disease course. Tremor is often considered a distinct symptom that does not always correlate with severe mobility decline.
That is the conventional clinical wisdom. But the smartphone sensors are detecting something the human eye cannot quantify.
During a standard clinical exam, a neurologist can observe a tremor and rate its severity on an integer scale.
But the smartphone accelerometer captures data at 100 hertz. That is 100 precise measurements every single second for a full 90 seconds.
So it is capturing subclinical physiological signals?
Exactly. It quantifies the microscopic continuous variations in the amplitude and frequency of that tremor. That level of granular digital data provides a highly sensitive early signal of rapid biological decline.
The researchers also noted that reaction time features were frequently selected by the predictive models, especially when patients were assessed in the unmedicated state.
Yes, that likely points to underlying levodopa-sensitive attentional mechanisms in the brain that begin degrading very early in these fast-progressing patients.
So the technology is clearly highly sensitive, but we need to connect this predictive capability to the real-world problem we mentioned earlier, the issue of failed clinical trials.
This paper provides a very clear example of how heterogeneity distorts trial outcomes. As we establish, this digital sub-study was part of the broader exenatide phase 3 trial.
And the overarching result of that parent trial was negative. Exenatide did not show an overall benefit in slowing motor progression across the general trial population.
It appeared to be a failed trial. However, when the authors looked specifically at the data-driven fast progressor group, they found a stark anomaly. The fast progressors who received the experimental drug actually worsened significantly more than the fast progressors who received the placebo.
They worsened significantly more. The data shows the fast progressors on the placebo experienced a mean clinical worsening of about 6 points over the 2 years. but the fast progressors receiving the drug worsened by about 14
A
points.
difference of about 8 points.
That is a very large divergence. A 14-point drop on a motor scale represents a meaningful loss of functional independence for a patient. So how should we interpret this? Does it suggest the experimental drug is actively harming this specific subgroup?
The authors are extremely careful to emphasize that this is an exploratory analysis. It does not definitively prove the drug is harmful or toxic,
I see.
but it perfectly illustrates the profound danger of trial heterogeneity. If you combine fast progressors and slow progressors without identifying them at baseline, your results will be heavily distorted.
Let us explore how that distortion happens mechanically in a trial.
Imagine a scenario where a drug is actually effective, but simply due to random chance, a larger number of unrecognized fast progressors are assigned to the active treatment arm, while more slow progressors end up in the placebo arm.
So the drug is battling a much more aggressive disease state from the beginning.
Exactly. You might analyze the final data and conclude the drug failed, completely unaware that it was fighting an undetected, rapidly progressing subgroup.
And conversely, the slow progressors on the placebo arm in the specific cohort barely worsened at all. They worsened by less than 1 point on average over 2 years.
Less than 1 point. If your trial population is saturated with those slow progressing patients, you can never demonstrate that your drug slows the disease because the disease is barely moving in the first place.
Which proves that conventional baseline clinical stratification is simply not robust enough. We must have data-driven, objective stratification.
We absolutely need to know who is on the fast track before administering the first dose of an experimental therapy.
And the smartphone application provides that precise stratification. But deployment is another hurdle. We have to consider patient compliance. Asking patients with motor symptoms to complete digital tests repeatedly at home often leads to high dropout rates in these digital studies.
Patient fatigue is always a concern. However, the compliance data in this study is actually very encouraging.
The protocol required participants to perform the 7-minute battery up to 3 times a day for a full week, following their standard 6-month clinic visits.
That is a significant time commitment for a patient. Yet, in the first week, about 80% of participants completed the assessments.
And about 76% met the strict compliance threshold for the analysis.
Exactly. Naturally, adherence decayed slightly over the 96-week study period, dropping to about 58% by the final week.
Which is expected in a 2-year longitudinal study.
It is, but the end-of-study questionnaires revealed excellent user acceptability. About 92% of the participants rated the application as easy to use.
And crucially, about 96% of them supported integrating this exact tool into their future in-clinic visits.
They clearly understood the value of providing their neurologists with this objective data.
Now, the researchers also wanted to test the real-world viability of this tool. In a real clinical trial, you will never have perfect data collection. Patients might forget their phones or refuse the digital test on a given day.
So they ran a digital dual pathway simulation.
They compared the standard clinical approach against a mixed digital approach.
Yes. In the mixed approach, participants who successfully provided the smartphone data were classified using the advanced digital predictive model. Those who lacked the smartphone data were simply classified using the traditional clinical model.
So they utilized the digital technology where it was available and fell back on the standard clinical exam where it was missing.
Exactly. And even in that mixed, imperfect scenario, the digital protocol increased the overall predictive accuracy for the entire trial population to about 80%.
About 80% accuracy in a simulated real-world deployment. That is highly actionable data.
It firmly outperformed the strictly clinical approach.
This highlights the immense scalability of the technology. Neurologists do not need to order a specialized MRI. They do not need to perform a lumbar puncture for prognostic biomarkers.
No specialized equipment is required.
You just use the standard sensors already built into the device the patient brings to the clinic.
It is highly accessible. However, we must responsibly acknowledge the limitations of this specific study.
Absolutely. The sample size of 98 participants is relatively small for a machine learning analysis.
It is. Furthermore, the cohort lacked significant diversity. About 95% of the participants self-identified as white, and about 78% were male.
They were also drawn exclusively from 3 hospitals in the United Kingdom under a very specific trial protocol.
Which means extensive external validation is required. We need to evaluate this algorithm across independent, diverse clinical centers globally before it can become a universal standard of care.
And requiring a smartphone and wireless internet could inadvertently introduce selection bias, potentially excluding patients in regions with limited digital infrastructure.
That is a valid concern for global deployment.
Despite those limitations, the immediate implications for our field are profound. So, to provide the practical summary once again for our listeners, Parkinson's disease progression is notoriously unpredictable, but combining standard clinical assessments with a 7-minute smartphone application allows neurologists to accurately identify fast motor progressors from day one.
This scalable digital framework provides reliable baseline stratification.
Which will ultimately allow us to design smarter, cleaner clinical trials, prevent effective drugs from being discarded due to hidden heterogeneity, and move us closer to true personalized medicine in daily neurology practice.
It is a remarkable advancement for the field.
It is. But the sheer precision of these standard digital sensors leaves us with a provocative final thought.
It certainly does. If the basic accelerometers in a standard smartphone can detect the subclinical microscopic signatures of rapidly progressing Parkinson's disease in just 7 minutes of active testing, what other health markers is the device silently capturing?
Exactly. Could the way a patient naturally types a text message, scrolls through a web page, or simply walks with their phone in their pocket eventually provide a continuous, passive early warning system for their neurological health?
Operating entirely in the background of their daily life.
It is a fascinating possibility for the future of neurology. Thank you for joining the weekly Neurology Deep Dive.
Thank you for the discussion.